All right. Thank you all for being here. Thank you to the organizers. I've really been enjoying my time here, and I'm looking forward to chatting with all of you about the gut microbiome. So within the concept of breathe, it might feel like a little bit of a stretch, but microbes respire. They're going to do anaerobic and aerobic respiration. And so I spend most of my research thinking about their fermentation, so those anaerobic components. And for those of you that join us at the pubs later, that beer is going to be also facilitated by microbes, so we can appreciate the importance of those. So fueling gut microbes. My research focuses on how diet affects intestinal microbes and health. And my background and training are in nutrition and gastrointestinal physiology. So I'm actually a registered dietitian. And so the way in which I approach research questions is going to be through a very translationally relevant lens, where I'm interested in providing foods in amounts that people would consume them. So a handful of walnuts, for example, a handful of almonds, a half a cup of broccoli, and so forth. And so to back up just a second and make sure we're all on the same page, I'll study the microbiota. So that's the collection of trillions of microorganisms that reside within the gastrointestinal tract. I also study the microbiome. When you hear that term, it is actually looking at all of the genes present. So it tells us what microbes are there and what can they do. And they're there in impressive amounts. The back of the envelope calculations originally were 10 times more microbes than human cells. It's actually more like 1 to 1. But their genes are exceptionally more relevant, 100-fold at least more genes than that of the human genome. And again, because I'm interested in diet, we can just look at their ability to break down something like fiber, which we cannot break down. We only have about 17 enzymes in our body that allow us to break down carbohydrates and sugars. They have about 17,000. So this microbial organ is what I'm interested in. And it's really been over the last decade or so that we've had the tools and techniques, the molecular sequencing capabilities and the computational approaches to really dive deep and understand that. And with regard to the health outcomes, my primary interest lies in metabolic health. So thinking about things like adiposity and insulin resistance. I do some work on the gut-brain axis as well. I do want to briefly mention that I also do a bit of work in exercise and just physical activity, and highlight some of the work that I've been able to do with some of my colleagues at the University of Illinois, who even have connections all the way back to Loughborough. So Dominika Pinjas actually did her doctorate here, finished in 2015 within the sports exercise within your group. And so these individuals bring their expertise. Jacob Allen did an exercise intervention that I was involved in. Nick Bird and his group, we published on resistance training and beef. And then Dominika, we just recently put out a cross-sectional analysis looking at accelerometry-based activity and associations with the microbiota. And so, and then my postdoc, Riley, has summarized a lot of this research in thinking about the connections between diet and exercise and some of the dietary patterns that athletes may want to undertake to optimize their performance and how that is interrelated with the microbiota. So to again think about how these things are interconnected, I want to certainly point out that both diet and exercise impact digestion, absorption, and transit time. Exercise type, duration, and intensity also impacts the gastrointestinal tract. So very intense exercise, you can certainly have issues with more rapid intestinal transit time, some damage potentially as well to the epithelial. And so these are important questions to consider within the sports area. For the purposes of this presentation, I will primarily be focusing on my area of expertise, which is the diet, but I'd be happy to dialogue about some of those more exercise connections during the panel and during the break. So the diet provides nutrients for gut microbes, and I've got some of those outlined here on this figure. Within the gastrointestinal tract, well, on our entire body, microbes are found. There are microbes that have adapted to live on our skin. There are microbes that have adapted to live within the very acidic environment within the stomach, but they're just fewer types can survive there. And their densities are much lower, 10 to the 7th. When you move more distally through the jejunum and ileum, you're going to get more dense and diverse populations, but the most dense and diverse microbial populations are going to be found within the colon. So about 10 to the 14th cells per grams. And I've got some of the commonly studied players listed there. And so I do primarily focus on bacteria, but archaea are present within the colon as well. I certainly have to mention that being at the University of Illinois, Carl Woese, who discovered that third domain of life. Our Institute of Genomic Biology is actually renamed after him. So Methanobrevibacter is one of the two most commonly found archaea within the GI tract. So again, thinking about diet and the microbiota, what I want to highlight here is that gut microbes will metabolize non-digested dietary substrates. They've evolved to be able to reside within our gastrointestinal tract. Again, I talked about 100-fold more genes than that of the human genome, and within carbohydrate metabolism, 16,000 to our 16. So I'm going to just highlight some of those, thinking about carbohydrates, primarily dietary fibers, because again, we don't have the genes to break down and absorb dietary fibers. There are lots of different types of dietary fibers, and that's why lots of different types of microbes can differentially utilize those. This fiber within an apple is different than the fiber within a whole grain, and so it will stimulate different microbes. Protein can also be used as an energy source for intestinal microbes, and then dietary fats and how that plays a role. So just to highlight some of these, when we think about non-digested dietary carbohydrates, so that would be your resistant starches, the dietary fibers, and prebiotics. You've heard that term. So those do mean different things, but different microbes, the microbiota, can differentially break those down. And when you think about this non-digestible carbohydrate fermentation, we're primarily thinking about the metabolites that are produced are short-chain fatty acids, acetate, propionate, and butyrate. You can also get gas production, and then the production of these short chains will affect water reabsorption within the colon. And so I don't like to say good and bad, but if we want to just dichotomize in that way, in general, the fermentation of non-digestible carbohydrates is good for the microbiota. The production of short chain fatty acids is good in general. We also can get dietary proteins that can escape digestion. We also have intestinal cells that get sloughed off and can be broken down by microbes. So amino acids are the building blocks of proteins, and you can see that the different types of amino acids will differentially result in different metabolites. So your aromatic amino acids like tryptophan and phenylalanine, those are going to primarily, the microbial metabolites would be phenols and indols. With your sulfur amino acids, you're going to result in sulfur compounds, dihydrogen sulfide, for example. All amino acids have an amine group, and so they can all result in that ammonia production. And then the branched chain amino acids can result in microbial fermentation in products such as branched chain fatty acids. So again, the point here, proteins, just like fiber, is an umbrella term. Proteins are made up of amino acids, and you're going to have lots of different metabolites that come out of that. Again, thinking simplistically, if we want to dichotomize, protein fermentation, especially within the colon, is bad. The associations between increased proteolytic fermentation and colorectal cancer can be quite high. So for example, dihydrogen sulfide. So in general, you want to see less of this. And so by providing adequate fiber so that microbes can utilize the fiber as a preferred substrate, that would be better. Some microbes can task switch. They can do both proteolytic, so both protein fermentation and carbohydrate fermentation. So if we provide adequate fiber, we can reduce the production of these protein fermentation end products. All right, what about fats? This other macronutrient we see on our nutrition facts label. Here I've got a schematic of the liver, the gallbladder, the stomach, and the intestines. And we make primary bile acids. So the liver converts cholesterol to trimethoxycholic and cholic acid. Those are going to be conjugated either with taurine or glycine amino acids, and then stored within the gallbladder and secreted into the duodenum in response to a meal. And so those primary bile acids have the main function of digestively helping to emulsify fat so that we can digest the fat and absorb it. Again, we saw the schematic earlier. There are lots of microbes within the intestinal tract. Bile acids are very cytotoxic. to bacteria. And so the types of bacteria that can reside within the more proximal intestinal tract have evolved to be able to deal with that. And so, especially when we get within the colon, some of those bile acids that do escape intrahepatic recirculation, microbes will convert those. They'll convert primary bile acids to secondary bile acids. And those secondary bile acids, again, are going to go into the category of bad. They are cytotoxic to colonic cells. There are strong associations with greater secondary bile acid concentrations in liver cancer. So we do not want great concentrations of secondary bile acid concentrations within the gastrointestinal tract. My research is also really interested in this evolving area too, where bile acids aren't just detergents for digestion and absorption. They actually will have different functions throughout the body. And because microbes will decorate them. And so you can have 100 different types of bile acids that are derived from both host and microbial formulations. And so those were doing some work specific to glycemic control. And so I've highlighted some of these different areas thinking about microbial-derived signaling and thinking about the importance of the microbiota. So to summarize in this first half, again, we've got the microbiota, this microbial organ within the gastrointestinal tract. And it is interacting with our dietary substrates and also the digestive components that we secrete to be able to absorb our food. And this is going to affect the pH of the intestine, for example. And so just to use this as an example, I talked about earlier the dichotomy. We do want to consume more fiber. Most individuals don't. In the US, at least, there's only about maybe 3 to 10% of individuals that meet that fiber recommendation per day. It's about 28 grams per day for women and about 35 grams per day for men. But when we consume adequate fiber, those microbes will make more short chain fatty acids. Again, we said those are more beneficial, and they'll make the pH of the intestine more acidic. Alternatively, if there's inadequate fiber, you can get more production of those proteolytic fermentation end products, and you're going to have a less acidic environment. So again, just thinking about the pH of the intestine, having a more acidic pH is beneficial because it can help reduce Campylobacter or E. coli. So thinking about those pathogens that cause foodborne illness, so the diarrheal diseases. So that's just thinking about pH. Inflammation, both locally and systemically. Short chain fatty acids are related to that. And hormonal secretion, the short chain fatty acids, for example, can stimulate secretion of different hormones like PYY, which is involved in intestinal transit and appetite. And we can even think more broadly. So I've mostly highlighted some of these within the gut health, but we are seeing increasingly that this has relevance, and Riley does a really great job summarizing this in this review, with these short chains and microbial metabolism having relevance for glycogen utilization and storage within the muscles. I had a great conversation earlier with one of your colleagues here thinking about the relevance of the microbiota for bone health. So we've seen a lot of a growing area cross-sectionally where the microbiota and greater short chain fatty acid concentrations has relevant for greater bone mineral density, but we still need a lot more intervention trials in that area. And then again, thinking about signaling through the gut-brain axis, and then where my work comes in as well, adiposity. And so just to highlight some of the work we've been doing in this area as well. So again, focusing on how diet affects the gut microbiota, we've shown in a clinical feeding trials that 1 avocado per day for 12 weeks affects the microbiota, enriching Calibacterium and Lactospira. These microbes can be perceived as beneficial. They've been related to reduce intestinal inflammation, and they also produce those short chain fatty acids. With the complete feeding trial with broccoli, where the only difference between everything that these individuals ate was broccoli was present or not for 3 weeks. This was done in collaboration with the USDA in Beltsville. We were doing some secondary analysis. So what does it do to the microbiota? Well, we saw an enrichment in Bacteroides. With oats and barley, some different types of grains, enrichment of Roseburia and Streptococcus, almonds and walnuts. So again, these nuts that are going to be rich in both fiber and unsaturated fatty acids, enrichments of Calibacterium and Roseburia. You'll see in both. Again, these are butyrate producing microbes. And so that has interest. And then to point out as well, within the walnut study, we reported the walnuts had reduced secondary bile acid concentrations. So by consuming walnuts, those individuals had lower concentrations of those pro-inflammatory cytotoxic secondary bile acids. We also showed a trending relationship for that in the avocados. So the avocados resulted in overall a slight reduction in both primary bile acids and secondary bile acids. And so we're subsequently following up on some of that work. And so just to briefly highlight some of that ongoing work on metabolic health. Again, I've noted this, but my group focuses on these diet health relationships. And so our interventions will range from everything from a sachet of fiber to everything an individual is eating or drinking for 3 weeks at a time, 4 weeks for when we start the avocado. Our health outcomes will range. Everything is simple from, point of care blood pressure to 6-hour mixed meal tolerance test, where we're sampling every 30 minutes following different meal challenges. So when we're providing different types of fats that we've been providing for 3 weeks to see how that changes the bile acids, and there's interconnections again between bile acids, glycemic control, and the microbiota. With the microbiome and metabolites, we're looking at lots of different things there. And this, of course, generates really large data sets. So we'll frequently have to utilize 12 to 90 plus processors at once so that you're not leaving your data spinning for a while. And that can happen. I just had a conversation with one of my PhD students the other day. She had reserved 12 processors, for example, but her code was wrong and it was just one. So we fixed that, right? We shouldn't be waiting a week for our microbiota data. It should be microbiota about a day. Microbiome, waiting a week might be realistic. So what are some of the things we're interested in? So just to again think about connecting this back to health, and where we followed up on that hypothesis generating. Some of the main things I'm interested in are lipopolysaccharides. These are components of bacterial cell walls. They can be pro-inflammatory. They can leak out of the gut and result in systemic inflammation. Secondary bile acids. So you've got kind of the bad players up here at the top. At the bottom, we're studying foods that have fiber. So again, these beneficial components that can be utilized as energy sources for certain microbes that we previously studied in nuts, Raspberry and Calibacterium, going back in to see, do they actually produce these short chain fatty acids? But more importantly, let's move out of the gut. And so I should point out here, this work with the bile acids, Jason Ridlon is really an expert at Illinois in microbial biology so that he can look at all of these different bile acids. So we'll move this out of the gut and think about the liver. The liver is certainly a first pass area for much of the blood flow from the intestine. And so these byproducts can have both positive and negative effects on inflammation and glycemic control as we move throughout the body. And so again, this is linking everything together from hepatic steatosis to the systemic inflammation and insulin sensitivity. And so hoping that complete feeding study has just randomized the final 30th participant for the walnut study. So hoping to wrap those up this year and then have these results from both an almond and a walnut study out hopefully next year. So just the last arm of research that I just wanted to very briefly highlight to show you again, what are some of the applications of the microbiota research? So it's not just the microbes that are there, it influences health in the previous one, but we can also use those metabolites and microbes as biomarkers. So over the last 7 years, some of the area we've been particularly interested in is exploratorily, could bacteria be used as biomarkers for food intake? And a part of the reason we're interested in this is, has anyone here done a food diary before? Or a 24-hour recall? They're quite laborious, right? And we think about this recall bias, especially as individuals age, or have had less sleep, right? I'm still a bit jet lagged, having arrived on Tuesday. I can't remember what I ate for breakfast yesterday, right? So when we can have fidelity measures, compliance measures in these intervention studies, they can be quite powerful. And so can bacteria be used as biomarkers for food intake is where we started, and we're subsequently moving out to health. So basically, just high level, you can do this by doing some feature selection for those, just create a compact set of microbes, different advanced analytic approaches like random forest to select what are the most relevant features and classify. And then of course, because this is quite exploratory and was a novel area of research, we spent a lot of time validating it. Are we predicting the food, or are we predicting that they participated in a study? And so that's where Qing Zhu, my friend and colleagues area of expertise at the University of Illinois, but I'm happy to say we were able to take the microbiota, the 16S, and predict food consumed. And so the level of accuracy was quite varied, though. So you'll see that it ranged from as high when we collapsed the oats and barley together at 96% to as low as 17% for broccoli. That's not useful. Less than 50% is chance, right? This is not useful information for the broccoli consumption. But the fact that it could predict between the walnuts and the almonds with quite high sensitivity, these are both nuts, that was quite promising. And part of the reason for that was because the background diets for both the walnuts and the almonds was identical. And so that gave us that additional statistical power to be able to detect that. And then we subsequently followed up with that looking at metabolites. And so again, there was a range of the efficacy for these. What we found for metabolites, so again, thinking about those short chain fatty acids, the phenols, the indols, et cetera, we had about 300 metabolites. They were not useful for predicting fecal metabolites for avocados, broccoli, or the grains. They were very close to chance. And so we really only published on the relevance for the walnuts and the almonds for the metabolomics. And I thought I had the references on there, but both of these were in the Journal of Nutrition. And so some of our ongoing work that we're about to submit are taking this again one step further. So using the metagenomic data and then multimodal data, so collapsing all of that together. And so those are about to be submitted for publication now. So again, hopefully those will come out in the next year or so. But with what we found just briefly, the multimodal, when you collapse those different data sets together, and you can create powerful models that can select and get higher predictive power. So for example, the metagenomics was quite useful for the broccoli, but we hadn't been able to find an adequate signal with the metabolites on their own. So I hope what you found in this presentation today, the key takeaways would simply be diet affects the microbiota and metabolome, that bacteria and metabolites can be used as biomarkers of dietary research, and that there is really a lot of ongoing research on microbial underpinnings of health and microbial biomarkers. So it's an exciting area to be involved in, and I think will keep me busy for quite a while. So thanks so much. We'll certainly acknowledge some of my collaborators here, the team that helped with the avocado study, and I will look forward to the discussion during the break and also during the panel. So thank you for your time and attention.